All right, so death in the CNS. So we talk about different types of death. Neuronal degeneration, specific result of sublethal injury, may progress to necrosis if the insult is not removed. So going back to just basic gen path principles, severity and duration of injury determine how severe the response is. Degeneration being the least severe manifestation of injury. When I use this term Malarian degeneration, this is a very specific type of degeneration that we see in neurons. A Malarian degeneration specifically refers to degeneration of neurons that have undergone focal axonal injury, such as that caused by a disc lesion, focal cord concussion. So when I say degeneration in the spinal cord, I'm referring to Malarian degeneration, which refers to if you have a compressive lesion right here, the stuff that is distal to the lesion will eventually die back. This is Malarian degeneration. Axonal change. The cell body will be fine. So this will contract. It might do a little bit of sprouting, but then the cell will live. Injury to the cell body of the neuron kills the whole thing. But if the neuron loses part of its axon, it can often survive. Necrosis refers to actual death of specific cell types. So when I say necrosis in the CNS, I'm often talking about usually individual cell types. And obviously the susceptibility to these insults varies by cell type, neurons being most susceptible because they're terminally differentiated. They don't have mitotic capability and they're highly specialized. So in general, neurons are the most susceptible to pretty much every kind of insult, especially things like hypoxia, hypoglycemia, stuff like that, and then kind of reduced susceptibility of all these other guys. So with necrosis of just specific cell types, the key is that the architecture of the tissue is retained. So you're not going to see this grossly. You're not going to see neuronal cell death grossly. You won't see changes until you start killing those support cells and losing the structure of the brain. So when we use the term malacia actually means softening. So this is referring to the grossly visible change that we see in the brain when there has been necrosis of multiple or all cell types. Since there's not really any structure or stroma, the brain just kind of falls apart. So that's malacia. It refers to pan-necrosis of all the cell types, not just the neurons and or the oligos, but everybody. As soon as you start losing astrocytes, they're providing the structure mainly of the brain. There isn't much, but what is there is provided by primarily astrocytes. So once you start losing them, the brain just kind of falls apart and you get this grossly visible softening. Again, pan means all. Pan necrosis means necrosis of everybody. All right, so necrosis versus malacia. Necrosis is selective cell death, usually neurons, but depending on the nature of the insult and the duration, this can progress to the death of multiple cell types, eventually reaching the stage of malacia. Once it's grossly visible to our eyeballs, then it's malacia. So if it's microscopically visible only, not malacia, we just call it necrosis. Malacia refers to this grossly visible tissue softening. Here it is. This lesion, do you think it's more gray or white? Can you tell? Kind of both. Here, you always have a contralateral for reference. So this part of it is white. This part of it is gray. It looks like we might have some early softening up here at the gray-white junction. That might be blue. So again, this type of necrosis is loss of architecture resulting in grossly visible softening. Okay, so we left off talking about death. This is my mystery neuro baby. His name is Voodoo. We euthanized him a couple weeks ago. He was 18 or 19 months old and had mystery neurologic disease that we still don't know what it is. So he may show up later in a case if we ever figure out what was going on with him. All right, so we talked about this. Any questions about what we started to cover on Friday? The difference between degeneration, sublethal, necrosis is lethal, death of specific cell types, usually neurons because they're most susceptible. And then malacia is pan-necrosis, all or multiple cell types. So you get grossly visible softening. Here's what that looks like. Single cell necrosis was not going to be visible grossly, only microscopically. You got to get down there and see that actual individual neurons. So this is a healthy neuron. He's got a nice happy nucleus, lots of cytoplasm. This purple stuff is missile substance. It's the endoplasmic reticulum. These neurons are shrunken. They are angular. They're hypereosinophilic. They've lost all their missile substance. There's no purple anymore. Chromatolysis. Chrome meaning color, so you've got lysis of all the color. So we colloquially call these guys red deads. They are red dead neurons. The nucleus, so compare these nucleus. This is pycnotic, shrunken, angular. This one's fading, falling apart. And then Malacia, grossly visible. So at this extent, you've got death of multiple cell types. You've lost the actual architecture of the tissue, and then it becomes grossly apparent as visible softening. Okay, so selective neuronal necrosis. What kills neurons? This is a short list, but basically everything. They're the prima donna cell of the nervous system. We talked about the other day how they're terminally differentiated. They're very specialized. They have high metabolic requirements, and all of this makes them very susceptible to many different types of injury. And a lot of these are pretty obvious. We're going to talk about thiamine deficiency a lot today. Infection we'll talk about Tuesday and Friday. And then I wanted to talk a little bit about this, excitotoxicity. Glutamate is your major excitatory neurotransmitter. But if you have too much glutamate, it's actually toxic to cells. So they think this may be the mechanism behind neuronal damage in things like seizures where you have abnormal firing. But this is a common mechanism of death for neurons. It's too much excitatory neurotransmitter. It's like too much coffee and they just like, you can't handle it. And then loss of stimuli or inputs. So once you get neuronal death and you start losing inputs to other neurons, like if the neuron above you dies, the one that's giving you your signals, and you're just hanging out like, hey, where did my signals go? Oh, well, I guess I'll die. So loss of input, either afferent or efferent, can also cause death in neurons. So what kills neurons? Everything. Malacia, this is one of the most common lesions you're going to see in the nervous system because the structure of the nervous system is so jello sponge, right? There's not a lot there to give it structure. There are no resident fibroblasts in the brain, except in the meninges. So there really isn't much there. So once things start to go, they really start to go. And you end up with softening, grossly visible cavities once it's progressed and severe. This is a non-specific change, just like necrosis in other organs. It's not associated with a single etiology. It's rather the end result of many different types of insult. The distribution might give you a clue to the etiology. Is it targeting the gray matter or the white matter? Is there some selectivity, or is it just everywhere? Is it the brain, the cord, or both? For lesions that are bilaterally symmetric, definitely that should cue you thinking about things that are systemic, especially toxic and metabolic. But note that not all systemic lesions present symmetrically. But if you see symmetry, you should think this is a systemic, probably toxic or metabolic thing. Resolution of Malacia, just like in other organs, you get removal of dead cell debris by phagocytes. Microglia, remember, are the resident phagocytes in the brain when they become activated and full of myelin. All these arrows are pointing to activated microglia that we call Gitter cells. I don't know why, probably some human guy naming them after themselves. I call them Gitter-done cells because they're getting it done. So all this vacuolation, all this foaminess is myelin. They're primarily digesting myelin, so they get very, very big and fluffy looking. And then this often leaves a cavity of some size if it's small. The remaining cavity can fill with a glial scar, which is composed of hypertrophic astrocytes. These are them. Usually astrocytes look more like this. They're just a big nucleus and you can't really pick out a lot of cytoplasm. These guys have massive, massive hypertrophy of their cytoplasm and also their processes, which I'll show you on the next slide. And these are essentially, they take over the function for fibroblasts since there are no fibroblasts in the brain. They make their big processes and they reach out and kind of form this network that makes a scar, what we call it a glial scar. And then for large cavities, obviously a glial scar is not going to be able to fill this. So these will glial scar around the edges of it, around around the edges of the cavity, and then the remaining will go with CSF. So here's what glial scar formation looks like. These are healthy, happy, quiet astrocytes, not super visible, not very much cytoplasm, a couple processes. Once they get reactive, they start to hypertrophy, and then they will form this kind of mesh or network that fills in defects of the brain. All right, symmetric lesions, you should always be thinking something systemic. A couple examples, this is Polioencephalomalacia in a horse. It's caused by yellow star thistle toxicity. You guys see the lesion? Oh, so there's arrows. You can see it in this one right here. So it's bilateral, it's symmetric. It is Malacia. Here is a histo-view of a symmetric systemic lesion. So we're in the spinal cord here. There's the white matter. Here are the gray matter horns right here. Central canal. You guys see the lesion? So this is normal gray matter. This gray matter is too pale. It's got little holes in it. And here's the other side. This is bilaterally symmetric. See it? Two circles. This is called focal symmetrical Polioencephalomalacia, selective to the gray matter of the spinal cord, and this is selenium toxicity in pigs. Right, and one more. There, a lot of these disease syndromes have focal symmetrical in the name. So focal symmetrical encephalomalacia in use. This is a classic finding with clostridial enterotoxemia, so clostridial toxins through the blood, absorbs systemically and produces very characteristic symmetric malacia. All right, so polioencephalomalacia, we talked about in general. This means necrosis specifically of gray matter of the brain. These are the three major causes in animals. Definitely know those. However, we also use this term polioencephalomalacia to refer to specific diseases. So polio in humans, polioencephalomalacia, and then polio in young ruminants, especially seen in feedlot housed versus pastured. This is a deficiency of thiamine. The etiologic association with thiamine is not 100% clear, but we give them early supplementation and many of them recover. So there is a pretty tight link, but not all cases will have thiamine deficiency as the etiology. You can also present these with these other other etiologies. So thiamine is important because this is a substance produced by rumen microbes, and it's actually a cofactor for glucose metabolism. And as we know, neurons require glucose. They can't function off, ketones or other substitutes. They have to have glucose. So if you don't have thiamine, you don't get enough glucose, and your brain suffers. The key to this being produced by rumen microbes is ruminal acidosis will often disrupt your microbial flora in your rumen, leading to secondary thiamine deficiency. So ruminal acidosis is actually a predisposing cause for polioencephalomalacia in ruminants. So thank you for the fourth last question. That's a question. Water deprivation, excess sulfur in the feed. So these sulfur actually cleaves thiamine. So a lot of these etiologies do have something to do with thiamine. And then thiaminase, if you see ACE, you know what that means, right? That's an enzyme. It's going to cleave something. So this is an enzyme that cleaves thiamine, and it's found in plants. Another side note, carnivores, especially wild fish-eating carnivores like foxes and mink, they get this condition, which is essentially polioencephalomalacia, but we call it chastic paralysis. And that's due to high amounts of thiaminases in fish that they eat in all fish diet and are not being supplemented. They often present with the same syndrome. Anybody know what this posture is called clinically? Opisthognose. So these are both opisthognose. It's arching the head over the back. All right, so reduced thiamine, decreased available glucose for neurons, so you get death of neurons followed by more susceptible glia. So this will start as selective neuronal necrosis. It'll progress to become visible malacia. So here we've got a cross section of cortex. These arrows are pointing to these kind of tan to brown softening spots. They are right along the gray-white junction. Here's a cavity, so this is a more severe area where we've actually lost tissue, and there's a cavity there. You're not usually lucky enough to see cases this severe. Usually it looks like this, which I have to imagine that you guys would have a hard time telling that from normal, because I have a hard time telling that from normal. The key here is, see this kind of translucent line that goes through the gray matter? And the gray matter is a little bit thin, a little discolored, very subjective, difficult to see. Here's the fun part: put it under UV light, and this lesion, if severe enough, will autofluoresce. Just that. It has to do with the metabolites in macrophages cleaning up all the myelin and debris. So the reason this follows the cortex at the gray-white junction is this is called laminar cortical necrosis. So microscopically, the cortical lamina are the arrangement of those neurons, and you're getting laminar necrosis of lamina. So it's like a whole strip of gray matter is undergoing necrosis, and the gray matter just goes flowers. Secondary consequences of this: lots of brain swelling, edema, herniation. Yes. Questions on polio? Also causing polio, salt toxicity, aka water deprivation. It's called both because it can be due to either. Direct mechanism is more common in ruminants. This is usually cattle given a salt supplement after a period of restriction, and they just, like me with chocolate, they just like can't stop themselves and just, oh, I'll eat the whole bag of chips. And then they salt toxicity themselves. This can also be caused by high salinity in your drinking water. So you can actually stand a lot of salt in your feed or as a supplement, as long as your drinking water is low salinity. But if your drinking water is high salinity too, you can't get away from the high saline. Indirect mechanism, this is more common in pigs. So this is too much salt only, respectively, considering you're not drinking any water. So they might be eating a normal amount of salt, but since they don't have access to water, it's too much salt. So this is more common in winter. Usually water sources freeze over and people don't have heaters or don't check on them, and the animals lose access to water for a few days. Then it warms up, they regain access, and they gorge themselves with water. So the common pathogenesis of both of these is that dehydration, whether from too much salt or not enough water, causes your brain to slowly become hyperosmolar to match your plasma, right? And this is done slowly by astrocytes who are forming your blood-brain barrier. So it takes time. And then you've got a hyperosmolarity in your brain. You've adapted to the high salt, low water conditions. And then suddenly you get as much water as you need. And your brain doesn't have time to adapt. And it's hyperosmolar. Water follows salt. It goes into your brain, and your brain goes. So cerebral edema, and then Polioencephalomalacia. That makes sense? This is what it looks like in pigs. They call this dog sitting. So this is a common presentation in pigs. It's pretty adorable for a clinical sign, as those go. You'll recognize this picture from before. This is the pig brain with salt toxicity and severe cerebral edema. You know, it's got edema because you've got herniation of the cerebellum here of the foramen magnum. It's coned. So this eventually will cause brainstem compression and death if they don't die from the laminar cortical necrosis first. In pigs, another classic lesion we also see is this eosinophilic perivascular cupping in the brain, right here. So here's a vessel. All of these around it are eosinophils. We haven't talked about cupping yet, but this is basically normal cellular migration out of the vessels in the brain. They come out of the vessels as they do anywhere else, and then this dense tangle of neurites and axons and processes kind of prevents their movement, so they end up forming a cup around the vessel. It's like a clogged freeway exit, basically. And remember in GenPath, I said pathognomonic lesions are rare. You should really pay attention. This is one. If you have a pig, polio, and eosinophils in the brain, you have salt toxicity for sure, 100%, 99.9%. All right, questions on polio or salt? And then let's remember the third one. Lead. Salt, thiamine, lead, polio. Yep.
The hyperosmolarity and like rapid dehydration or rehydration also affect like dogs. If you find that a dehydrated dog, should you not put them on fluids?
I would say no. This is more of a large animal problem. I won't say it's impossible that an animal or that a dog or cat or other small domestic house pet, anything's theoretically possible, but usually this is large animals, ruminants. When I was in vet school, there was a big mini pig craze, and all the mini pigs, because people don't know how to take care of pet pigs, right? They know how to feed and water a dog for the most part, or a cat, I would hope so. But they would come into the large animal, or they would come into the small animal hospital, and they're like, this isn't a dog or cat, I don't know what to do with this. And they'd send it over to the large animal hospital, and they'd be like, this is too small, it won't fit any of our equipment. They'd send it back to the small animal hospital. So yeah, more of a pet pig thing, but it's going to be a pet. Other questions? All right, leukoencephalomalacia, literally white brain death. So this is selective white matter necrosis in the brain. Pause. Seen this happen with a Labrador that went to the beach and drank too much seawater. That's really sad. Thank you. So yes, can happen, theoretically possible, not common. If anybody could do that, it'd be a Labrador.
What about a dog that eats Play-Doh?
Does it have access to water?
I don't know. Well, I mean, it gets.
Yeah, you can withstand a lot of dietary salt as long as you have access to fresh water. You guys are like trying to think of scenarios now, like what if this? Okay, leukoencephalomalacia. These are the demyelinating diseases as well as this classic syndrome, mycotoxic leukoencephalomalacia of horses. I famously hate horses very much, so it's the worst thing. This results from prolonged feeding of moldy corn. So usually in winter, when there's not as a lot of fresh stuff happening. So this is moldy corn. So usually in winter, especially it's like here where you get mold very rapidly. And this is due to toxicity of Phlomonas B1, which is produced by Fusarium. It's a common saprophytic mold. It's everywhere. And what this does is it blocks synthesis of membrane lipids. So this results in damage primarily to myelinated axons, the axons themselves, the myelinating cells, anything where your membrane is really important. So clinically, they'll present with sudden onset of weakness, staggering, impaired vision, basically cortical signs. There is no recovery once this sets in and you've got clinical signs. That's it. This is a fun quote that I found from JPP, which is one of our major pathology texts. Chronic cases with static signs are dummies. I'm like, how is that different from a regular horse? Pathology is selective, irregular. This is systemic and toxic, but it's asymmetrical. So this is one that's not, you can't count on the symmetry to give you a clue, but it does target the white matter. And if you have white matter lesions in a horse, you should think Phlomonas and mycotoxic. Here's what that looks like. This is the picture from earlier. It's hard to convince you that this is white only because it's actually kind of bleeding into the gray matter, but there is this kind of line of demarcation here, and it is predominantly white. Here's one that's more subtle. So this is these three are all fixed specimens. This is a fresh specimen. So we don't see very many fresh pictures of brains because usually we just throw them right into formalin. We're scared they're going to disintegrate. So this is a fresh brain. And the primary finding here is this yellow to brown discoloration right here. And then all these little pinpoint red things that are basically lining the gray-white junction. So this is hemorrhage. There's a little petechia in here. This brown is Malacia. So you could see the way it's doesn't really go into the gray matter here. This is a much better example of selective target white matter. All this yellow discoloration here. There's a little cavity or a little pocket. Here is a big cavity. Amazing, this animal was alive as long as it was. That's a very, very severe lesion. All of this is a big malacic pocket. Here's another fixed specimen. I like this one because you can really appreciate how it pretty much spares the gray matter. You can follow it all the way around. However, the white matter is reduced in thickness and size. So you've actually lost tissue. It's discolored. It's not white anymore. It's kind of brown. It should be this color, this like very bright, shiny white. Instead of it's this normal brown. You can see it's kind of sunken. If you look at the contour of this, it looks kind of like it's kind of sunken in. It's sunken. There's a cute cavity. Oh. And then what is all this very bright white, pretty stuff? Mineralization. Is it dystrophic or metastatic? Yeah, it is. I love you guys.